Hi, I'm Eric Schock with the Indiana University School of Medicine. As of September 2009, more than 40 of our scientists have received research grants from the federal stimulus program, and we thought we'd talk to a few of these scientists about their work. Carmen Yoder is a radiologist whose specialty is using modern imaging techniques to study brain activity. She will be using PET scanning to study how a brain chemical called dopamine affects the pain and other problems of fibromyalgia. Her $237,000 grant will enable her to hire a new research assistant for the project.
Over the last couple of years, there's been increased evidence suggesting that dopamine may mediate some of the symptoms that fibromyalgia subjects perceive. Fibromyalgia subjects also complain of cognitive problems, and dopamine is definitely involved in cognition. So there might be a link between dopamine and pain sensation. There also may be another link between dopamine and cognitive problems in this population. So it's of interest to us because fibromyalgia subjects are often prescribed really addictive drugs for pain-killing purposes. And we want to try to circumnavigate the addictive liability, but to do this we have to understand what's going on in the brain. So if we can understand what dopamine's doing and what's going on in dopamine in fibromyalgia subjects, this may be helpful to provide alternative therapies for people that are suffering from fibromyalgia in the future.
Mark Kaplan is studying a protein called STAT4 that's involved with the inflammation that occurs in such diseases as type 1 diabetes, arthritis, and colitis. Dr. Kaplan's $385,000 grant kept 3 of his laboratory staffers working, including Raji Muthakrishnan, who received her PhD from IU last year. Working with colleague Stephen Steiner, Dr. Kaplan will be studying children with ulcerative colitis and Crohn's disease to determine whether STAT4 can be used to develop a blood test for those and other immune diseases.
So being a central mediator of disease, STAT4 is a potential target for a large number of therapeutics. And if we could find pharmaceuticals that would be able to interfere with STAT4 function or interfere with some of the targets that STAT4 activates during the process of developing disease, those would be potential treatments for a lot of different types of autoimmune disease. And so there is a lot of interest in that.
So understanding how the T-cell development is regulated is essential for us to know how it can go wrong during disease process and how we can fix it if it goes wrong.
So some of our recent work has focused on isoforms of STAT4. And what we found is that one of these forms of STAT4 is more involved in inflammatory disease than the other form. So what we've been able to do so far is in patients that have either colitis or Crohn's disease, show that they have increased ratios of the inflammatory to the non-inflammatory form. And what we're hoping is that this may tell us something about the type of disease that the patient has, as well as potentially being a test to identify how severe the disease is and what might best be used to treat that disease.
Ruben Kapoor works with the rare bone marrow cells that produce the billions of red and white blood cells our bodies need every day. His work could help improve the life-saving cord blood and bone marrow transplants that are used to treat leukemia, lymphomas, and other diseases. Without his grant of about $419,000, Dr. Kapoor said he would have had to cut his laboratory staff by 3 full-time jobs.
So one of the major uses of, or one of the major usage of a system like this would be in a setting of cord blood transplantation. So we know that in cord blood transplants, the cells that we get from the cord blood are often less in numbers. And if one could expand these cells in large enough amounts, then we could perhaps use these cells for transplanting adult patients who need these types of cells. So in that setting, there will be a direct clinical relevance for this type of treatment.
Richard Day, a physiology researcher, studies how mutations affect protein activity, in particular a pituitary gland protein called PIT1. Mutations that block that protein's function cause serious problems for patients. Dr. Day's grant enabled him to add a summer research assistant.
Patients who have inactivating mutations in PIT1 cannot produce the cells that will make the hormones prolactin and growth hormone. When these patients survive, they have a syndrome called combined pituitary hormone deficiency, which means they're deficient in growth hormone and prolactin. So the patients will be of shortened stature, and because they're deficient in prolactin, they'll often have reproductive difficulties, but they survive. So we're interested in understanding how these particular point mutations in the PIT1 gene function to inactivate its activities in the anterior pituitary. The grant under the stimulus package has allowed us to hire an undergraduate student for the summer to work specifically on this project.
My name is Laura Weddis. I'm an undergrad biology major here at IUPUI. I work in Rich Day's lab here in the Medical Center on campus. I went to eventually work in a lab at like Lilly or Roche and work with couples that are expecting a child that may have, genetic disorders that run in their family, and I want to try and find a cure or help that process along. So that's what I'd like to do.
It's allowed us to accelerate the pace of research on the parent grant that would not have been possible without the stimulus money.
You can find out more information about research at Indiana University and the School of Medicine at our websites, www.iu.edu and medicine.iu.edu.